No one is immune from the ravages of time. The detrimental effects of aging are inevitable, right? But what if we could turn back the clock and replace worn out or damaged body parts with completely new ones? It may sound like science fiction, but researchers studying cell growth are currently working to make it science fact. Imagine a future where no one waits on an organ transplant list. If your kidney failed, you could have a replacement made to order in a lab. Using your own cells, technicians could build a new kidney using a 3D printer, and with this printed organ, you'd quickly get back to a productive life. Imagine a future where if you're disfigured in an unfortunate accident, a pill could change the electrical charge at the site of the injury, causing your toes to regenerate. In the future, if you suffer from heart failure, a new organ could be grown on a cadaver heart scaffold using your own stem cells. And with this new heart, you could get back to running. While these three futuristic treatments are only in the experimental stage, there is one science fiction sounding therapy that is already being used on humans. Take the case of Marine Sergeant Brian Smith, who was injured while leading his squad down one of the most treacherous roads in Fallujah, Iraq.
I was on a mission to get my squad familiar with the new area of operation that we were in. That entailed traveling down IED alley.
Smith's Humvee struck an IED, or improvised explosive device.
We were just traveling, and then all of a sudden we got hit, and I believe I went out for a second.
A piece of shrapnel sliced through the tricep and bicep of his right arm, severing a major artery and leaving his muscles in tatters.
And I realized I couldn't move my right arm. And at first I thought it was gone, but I could feel that it was still there, and I could feel the blood, my blood from my artery pumping into my hand as I reached over.
Sergeant Smith was airlifted to Germany for treatment. Back in the US, rehabilitation of his arm left him weak, unable to assist with the care of his 9-month-old son.
He was go, 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 go. One of the issues is trying to care for him. I couldn't pick him up if he's squirming because I only had one hand. Couldn't change him because, you know, you want one hand to try to keep the kid still.
Brian compensated for the weakness by using the strength in his back. 2 years later, he was able to join the Bangor police force, but that weakness in his arm was always on his mind.
My concerns about my arm was anything that required pulling. You're trying to take somebody into custody. Sometimes you have to pry their arm out. I'd have to do it with my right hand, but I knew I was now significantly weaker in that hand versus my left hand.
Brian heard about an experimental procedure at the University of Pittsburgh that uses an extracellular matrix to regrow damaged muscle tissue. A year after treatment in 2013, things were remarkably different with his arm.
The big thing is I was able to hold my child in my right arm longer than I ever have.
Brian had lost 1/3 of his bicep. With ECM, he was able to regrow almost all of the missing muscle tissue.
Today my arm feels great. My endurance is there. That was the big thing for me.
Now Brian can do his job with complete confidence and be the hands-on father he wanted to be.
Good evening. Welcome to our program. Thank you all for coming. Being forever young, the future and the promise of human regeneration. So without further ado, let's meet our scientists, beginning with the Director of Regenerative Medicine Research and Director, Center for Cell and Organ Biotechnology at the Texas Heart Institute in Houston. She also holds faculty appointments at Texas A&M and Rice University. Please welcome Doris Taylor. Our next guest is a professor in the Department of Surgery and Deputy Director of the McGowan Institute for Regenerative Medicine at the University of Pittsburgh. Dr. Badylak is a past president of the Tissue Engineering Regenerative Medicine International Society. Please welcome Stephen Badylak. Also joining us is a principal investigator in the Tufts Center for Regenerative and Developmental Biology and research professor in the Department of Biology at Tufts University. She is also faculty in the EBICS program at MIT and a member of the Allen Discovery Center at Tufts. Please welcome Dany Spencer-Adams. Our final guest is an associate professor and associate director of the Nancy E. and Peter C. Meinig School of Biomedical Engineering at Cornell University in beautiful Ithaca. He's been recognized with awards given by the Society for Heart Valve Disease and the Biomedical Engineering Society. Please welcome Jonathan Butcher. Welcome. So we want to start tonight's conversation by asking each of you, beginning with Dany, just to give us a little bit of a primer, if you will, on exactly what you're working on.
Well, I actually came to study regeneration through studying generation. And I believe you're about to see a movie that I made showing the generation of a face in a frog embryo. So there you're looking at a frog embryo right now. And that bright spot coming up over the horizon like that is actually the central nervous system forming. And you're going to see spots that are making where the face is going to be. And what those spots are is they're a dye that's showing us where the cells are more negative on the inside than they are on the outside. So it's an electrical characteristic of the cells. And what we found by studying these developmental processes is that these electrical signals help the embryo know where to put things and, very importantly, how to shape things. And that turns out to be a very, very important aspect of regeneration, which is how do you get the proper shape for function.
And shape turns out to be important. We're going to come back to that. Stephen, how did you come to actually grow muscle tissue? Tell us about this.
Our approach is a little different to regenerative medicine, and as you saw in Sergeant Smith there, our intent is to provide the environment that the body normally uses to send signals to say, okay, stem cells, come on over here, form this particular type of tissue, or these cells should proliferate. In other words, it's sort of like the movie, you know, build it and they will come. Mm-hmm. So that's what we want to do is provide the microenvironment, what's called the extracellular matrix, and then let the body do the rest of the work. So it's a little different approach, but it seems to be working.
Doris, you have a different approach altogether. Tell us about that.
Well, heart disease is the #1 killer of men, women, and children. So what we've done for the last 20 years is work on ways to repair the damage in the heart. We started with genes, but what we realized is that you had to get lucky and pick the right gene, and that cells were little gene factories, so we moved to cells. But if you're going to put a cell in the heart, just like Steve said, it needs a right environment. And so it's probably true that we're never going to get enough cells to take a fully damaged, leathery, thin scar and make it into healthy heart again. So what we said is, wow, we need an environment. Let's take a heart. Let's give nature the tools and get out of the way. So today, I work on building a heart in the lab by taking stem cells and putting them in what we call a ghost heart, a heart where we've already stripped all the cells to let nature provide those cues to rebuild the heart.
Amazing, amazing. Jonathan, I think even more amazing, or equally as amazing, is printing a heart valve. How does one begin to even think about printing a heart valve?
Well, so I think I came into this approach as a traditional mechanical engineer. Like Dany, I heard a lecture on developmental biology of the heart, and this biologist proceeded to describe how the heart forms inside of a week in this animal. And here I am trying to engineer something to change a protein in months. And so clearly there was so much natural engineering going on that we didn't understand. And so I sort of shifted training into developmental biology, and then now in my current lab, we combine both of those sets of tools to be able to try to biofabricate these organs.
Well, let's get to the idea of actually how you are doing this and what this stuff actually looks like. So Doris, take us through the how to make a heart, if you will.
Well,
and you can do this at home, as I understand it.
That's right.
Right.
That's right.
It's available online?
There are blogs about how to do this online.
All right.
And I think that illustrates an amazing point, that that people really want to understand the science of this and the biology of this, and there's such an unmet need. I had a high school kid call me last week and say, our family's barbecuing a pig in the yard, and my family gave me the heart, and it's in the refrigerator. What do I do? I told her. I said,
tell us,
what
do we do?
So what do you do?
Yeah.
Well, if you're going to build a heart, you need cells,
okay.
but beating cells right here in a dish aren't enough to make a heart. You need a place to put those cells. So you need a framework or a scaffold. So what we did is we took a heart, a pig heart, or a human heart. We now work routinely with human hearts, and we wash the cells out. We use baby shampoo, literally, to wash the cells out of the heart. And you can see here on the video a pig heart in this case with no cells. So
what is that? Is that collagen? What is it?
It's the extracellular matrix, proteins.
Okay, proteins. Okay.
If you touch it, it feels a little bit like a Jell-O wiggler. And for those of you who aren't old enough to know what a Jell-O wiggler is, it's dehydrated Jell-O. It's actually pretty tough. But we can then put cells back into that heart.
Okay.
And when we put cells back in, what's so amazing is you put a blood vessel cell in, and it goes to the blood vessel. You put a heart muscle cell in, it goes to the muscle. We call it our smart heart. It knows what to do. Here, you see a heart where we've put cells back in, and it's pink now because it's beginning to be more muscle, and it's moving because you can't just put a stem cell in a heart and let it sit there. It has to act like a heart, look like a heart, and work like a heart to grow up and
to know what to do.
To grow up and become a heart. So it has to have a blood pressure. It has to have lungs and oxygen. This is a human heart in one of our bioreactors.
Well, what is that? Sounds
so
very...
a bioreactor is basically an artificial body.
Artificial.
It's got blood, it's got oxygen, it's got temperature, and it's got an immune system. So and you see here the plastic container, that's the bioreactor. Keeps it sterile, keeps it warm, food, clothing, shelter, everything you need to build a heart. So we put stem cells back in, and they mature over time if we give them the right cues. It's pretty remarkable. And you can do it with anything. Heart, liver, kidney, lung, pancreas.
Unbelievable. Stephen, tell us your story. How are you doing, what are you doing to get muscle?
Well, what we do is start with the matrix. And
again, with the matrix.
Back to the matrix, that's right. And it's a matter of, you can isolate every tissue in the body of every person in this audience has extracellular matrix around the cells. That's the scaffolding material. And in this video, what you see is Scott in my lab. That's a pig bladder that you can go to the slaughterhouse and pick up, and instead of them, it's a byproduct of the agricultural industry that would have been fertilizer or food or something else if we didn't pick it up. And we take away the muscle layers, and we're left with only the layers that we want. And so that's that thin layer that Scott was holding up there. And then we take out all of the cells. And what you're left with is this friendly inductive material. In Doris's case, it was a three-dimensional heart. In our case, it's this simple sheet of material that we can keep as a sheet. We could turn it into a hydrogel. We could turn it into a liquid. And you're looking at muscle cells there that were formed and it wasn't Sergeant Smith's, but one of our other patients in a recent study where we treated 13 patients like Sergeant Smith, all of whom returned to a quality of life where they could do the activities of daily living like he did. So basically what we do is change the default, that this material has the ability to signal the body to say, look, instead of responding to this injury by forming a scar, what we want to do is recruit the body's own cells to it and have them assemble, as Doris was saying, the way that they would normally, and recreate the tissue rather than just repair the tissue. So it's a little different approach. And like I said, none of us have the solution to all the problems, but we all get there a little different way.
Absolutely, unbelievable. Dany, you're using electricity. Is that fair to say? To make.
Absolutely.
So that sounds Frankensteinian, or however
very
you say
Frankenstein.
it.
I love the analogy. The idea is that by studying these electrical signals during development and seeing that they can be the signal that says, okay, you be a muscle cell, these cells start dividing, those cells stop dividing, that this is one of the ways that cells talk to each other. So chemicals is one important way, but electricity is another important way, and it's just the movement of ions. So everybody has heard of pH, which is, it's just a fancy way of saying the concentration of hydrogen ions. And hydrogen ions moving, that's a current, that's electricity. And we found that there are so many places in development of the embryo where those electrical signals seem to be, I hesitate to use the term, but master switches. They really do seem to be like you flip a switch and then a process gets started. And very importantly, it doesn't just get started, it gets controlled and it gets stopped at the right time. So you say, okay, well, will this work in regeneration as well? And have found that yes, again, in that situation, it's these very, very powerful signals that coordinate. They take care of everything. What you're seeing here actually is one of the really important stages in vertebrates, so things with backbones in regeneration of vertebrates, is that you have to have nerves there for the rest of the structure to regenerate. And what you're looking at here is a lot of nerves that have grown places where they don't normally belong. That's a close-up of a fin of a tadpole of a frog. And this is work by Dr. Douglas Blackiston. And what he did to get that to happen was he simply soaked the tadpole in serotonin, so the neurotransmitter. And what happens through a chemical signaling pathway is that the serotonin makes the cells in that fin be a little more positive than they would normally be. And that attracts the nerves. And so a place with no nerve suddenly has this huge invasion by all these nerves. Yeah. He's actually done some truly remarkable work where he has, it's a combination of transplant, but then looking at what happens to the nerves, and he puts a piece of tissue that would be an eye, and he puts it into the flank of another embryo. So it puts it in the wrong place. And he uses an eye because that will then send out a large nerve, the optic nerve. And sometimes that optic nerve actually finds its way to the spinal cord, and that eye can sense light. It can see. So the brain somehow knows that it's visual information, even though it's coming from all the wrong place. But again, it's this idea of being able to control and use those electrical signals to get what we need to happen, in this case, for nerves to grow. And in other cases, for entire limbs to reproduce themselves.
Show us what that's about.
Okay. So what you're looking at here is the back end of a frog. And if a frog loses a limb, what you see here is the one that is the left-hand side. That's a completely regenerated limb. So that was amputated up close to the knee there. And normally, if that happens, you get what's called a spike. And it's just it's exactly what it sounds like. It's a spike. But this was work of Dr. Aisun Tseng, who, when she was working with Mike Levin, and she treated that wound with a little cocktail. And that cocktail did the same thing. It made the cells change their electrical characteristics. And 6 months later, this grew back. So instead of getting a spike, she got a foot. And this is a foot. It has, you can see the blood vessels. It's got skin. It's got muscle. It's got bone. And very importantly, it has nerves. And it's also the right shape. And what you're seeing here is she's just poking that to show you that it's quite responsive. It has a very well formed and functional nervous system. So this incredibly simple message months ago, which was change your electrical potential, and this entire limb has grown back, and it's the right shape, and it stops when it's done.
Unbelievable.
It's quite extraordinary work.
Yeah, yeah, that is cool. So give us Jonathan, the how to print a heart valve for dummies, if you will.
Well, I mean, I think we're all aware of this new revolution of 3D printing, and there's these maker communities and maker spaces and all sorts of printers that are now entering, the community for the ability to make almost anything. And I say almost because making something that's completely living is very different than making something out of plastic. But in reality, anybody who has ever made a birthday cake can 3D print something that is as complex as a heart valve. I would say the way that we need to do it differently is because every patient has a different heart valve, and or heart, or whatever. But that geometric complexity is actually extremely important for that particular success of that particular graft for that patient. And so what we have to do is work with surgeons to get clinical imaging, which you see here, that defines the actual geometry needed to extract and then replicate in a 3D printer. Now, once you have that geometry, you can actually use computer algorithms to develop a deposition or printing path, and you can use a printer like the one you see here to essentially deposit material along those defined paths. Now, the challenge then becomes, well, what do you print? Because it's not going to be frosting or plastic. So we actually develop in our lab these materials. We could call them living inks or slurries, and they're actually comprised of biologically derived components of extracellular matrix, potentially from tissues like the ones that Doris and Stephen are talking about, but also we can make them from scratch. And the idea is that you can design point by point what material you want where. And also we can embed stem cells in those same environments. And the opportunity is that now you can put the instructions that you want to give cells in each location, so that in one location they make one type of cell, in another location the stem cells turn into a different type of cell, all the while achieving the geometric complexity.
Is that what that is?
Yeah, so that's a human, it's an aortic heart valve, a tri-leaflet heart valve, that we've manufactured. And you can see a couple different sizes that we make here, because one of the major clinical needs for this type of technology is actually the pediatric populations. In particular, for a scaffold or a graft like this that has the living capacity to grow. And so you can't really use a prosthetic for that. So there's really nothing other than this kind of an opportunity to go. So that's why we're pushing this envelope.
Okay, so one of the things that I think we have to talk about is the elephant in the room, which is genetics and genomics. And that's an inside joke. There is a paradigm shift here, because you guys aren't doing anything with genes. You're using like you're coming at it from a completely different angle. Tell me about the paradigm shift and how it happened and what it means.
Well, to me, genes, as I said earlier, if you're going to work with genes, you have to know the right gene, and you have to have a cell for that gene to work. Genes don't happen in a vacuum, they only work in a cell. So we said we can spend the rest of our lives trying to pick the right gene, figure out the right gene, or we can take cells, which are little gene factories, and let nature drive which genes by providing the right environment for those genes, whether it's for those cells, whether it's chemical, a scaffold, or whatever. Genes are the potential. Cells are the reality.
Okay. Stephen?
Well, I pretty much agree with Doris. I think I'm a little more positive on the future of using the human genome and our understanding of molecular biology for personalized medicine, because each of us has our own unique set of genes. And I think the more that we understand about your set versus my set, one particular therapy is going to work better for you than it's going for me. I think that's where we're going to really use that particular approach to regenerative medicine. But one of the things I find the most interesting about what we do, and what makes my job so much fun every day is, when we put an extracellular matrix either in a petri dish or in a patient, why do certain cells get attracted? Right. What signals in that matrix tell that stem cell that you should now differentiate into a bone or cartilage or whatever? And it gets down to the molecular signals that are there. So what are those signaling pathways? An interesting, here's a real life example. And without getting into too much detail, we now understand why can you take a pig extracellular matrix, put it into a human, and not have problems, right? Why doesn't it get rejected? So we're very interested in that question. One of the reasons is because the molecular makeup of the proteins and the other molecules that form my extracellular matrix and yours and a pig's and a mouse and probably a dinosaur is so similar that the recipient says, okay, you might not be me, but you're not so bad. I think I understand, what you. So we don't reject it, and then actually it breaks it down, and it's loaded with these signaling molecules. And so one of the things we found out in a recent study, because we were interested in the immune system, was what tells the immune system to accept and not reject? Well, the intracellular pathway involves the same pathway that is inhibited by non-steroidal anti-inflammatory NSAIDs, like Advil, Naprosyn, things like this. So what we learned is that if you put the patient on these non-steroidal drugs after surgery for pain, you're actually blocking a pathway that's important. So from that standpoint, it's really important.
All inflammation,
yeah,
Steve.
It is. Inflammation, or if you would, I'm even sorry that word exists because it implies a negative connotation, and without inflammation, none of us would be here.
Inflammation is nature's cue to say, I've got an injury, send me cells.
Yeah. So let's finish with one more comment, because Jonathan's an engineer, Dany's a molecular biologist, I'm a physician, and Doris is everything, but none of us have the answer. An interdisciplinary approach to these problems, using the molecular biologists and the engineering and such, is where the greatest advances are going to be made. One of our problems is that our method of educating is
siloed.
It's ancient, and it hasn't changed much. But we are recognizing interdisciplinary approaches to problems yield the biggest solutions.
Before we get too far away from the genome and genetics, is one reason we don't study it is because there are lots of people studying it, and there's doing tremendous, wonderful work, and that's not what we're interested in, but that's all being done really beautifully in labs all over the world. But the other part of that is it hasn't worked yet.
Nobody's taken that stem cell and turned it into a heart.
Nobody has said these genes are the things that give you this shape.
Got it.
And in fact, there are these people now work on things called gene regulatory networks, and these are genes that turn each other on and off and trying to figure it out, what is actually going on, who's the message passing from to, like a game of telephone almost. And you can have the gene regulatory networks, you could think of as a map, and they have, one map that has 50 different things in it gives you a tube that's a blood vessel. But if you have another tube that's for something different, the gene regulatory network could be completely different. There's nothing in there yet. We don't know yet how to look at that and see where a shape actually comes from. And that's because we haven't yet figured out how to bring in geometry and forces and directing forces and resisting them and generating them. And it just simply hasn't worked yet.
Don't you think nature has developed a lot of different ways, though, to create the same things?
Absolutely.
If you look at it, we have tubes in our blood vessels, we have tubes in our lungs, we have tubes in our kidney, in our bladder.
Tubes are great. We are tubes.
Everything, right.
A little more than that.
So the body has created, there are a number of different ways to create every single thing that exists. So it's not one gene, one endpoint. And I think what we're doing is saying, at least my view of it is, we're bypassing a lot of that, and we're saying we've got the shape. We're letting nature, we're taking advantage of what nature's already built with those cues already in it, and letting that drive what we decide to do going forward. So we can spend the next 20 years figuring out all those networks, or we can use what nature's built and go forward.
Stephen, did you want to add to that?
Well, you know, I was trying to figure out how to get this comment in, but maybe just give you an opening. The developmental biology, if we don't understand normal development, how do we expect to develop effective strategies for recreating normal functional tissue? So we have a couple of regenerative organs in our body. The liver is constantly regenerating by a mechanism that's different than the axolotl, but it's a regenerative. Our bone marrow regenerates all our blood cells every day, our skin, outer layers of our skin, and one or two other tissues. But through evolution, the rest of our tissues have changed to say we've sacrificed regeneration for healing, for scar tissue formation. And it's really interesting, I think, in developmental biology. If you look at a human fetus in the first trimester can regenerate all sorts of organs. And somewhere around 16 to 18 weeks of gestation, right when our immune system kicks in, when we develop this ability to fight off infection and to respond to injury, we lose the ability to regenerate. So what genes are being turned on, which genes are being turned off? And I think if we just would work together a little more, Dany, we'd get the...
But that's an important point, that there's a roadmap there for regeneration that could be understood better.
And you know, you can think about that even after we're born, though. If you look at a 2-year-old and they fall down and scrape their knee, it's going to get red, that inflammation's going to happen, and that's going to be nature's cue to say, send me more cells, and they heal that, but they probably don't have a scar for the rest of their life. 2-year-old gets a cold, it goes away in 3 days. They've got cells, and they've got potent, healthy cells that we don't have. When you're 52, 62, you fall down and you scrape your knee, the cues aren't the same.
There's a wonderful story about that, actually, that up to about the age of 7, and depending on who tells you the story, it's 5, sometimes it's 10. But when you're a kid, you can actually lose the tip of your finger. And generally, the standard procedure then is to take skin and wrap it over to prevent infection. But if you don't do that, there is a current called the healing current, and it's just positive charges leaving the tip of the tip of the finger, and the finger will regenerate. And it'll regenerate the bone and the skin and the muscle and the nail. And we lose that ability, you know, somewhere 7, 10, something like that. But what that tells us, and the liver and all these things, it tells us that our cells do know how to do this. And we can either...
cells can do that for 2 weeks after birth.
For 2 weeks. So all of our cells seem to know that it's in there somewhere. And whether we need genetics to trigger it, if we can help them along. I know that some people now, correct me, but isn't one of the treatments for losing a fingertip is to pack the wound with the pig bladder, right? Yeah, to provide the scaffold. Yeah. So there's learning how it's done. There's learning how we might be able to manipulate it to trigger it. Now, whether that's exactly the way it was done when the embryo was developing and turning into a fetus, or whether we do it by, you know, tricking it, turning on the wrong gene, but it has the right effect. That's where that turns into medicine. That's where you go to translation, and how do we don't care how it was done, we care how to make it happen again.
Let's talk about what are some of the actual short-term and near-term benefits of what we're talking about tonight? Where do we see things, if not right now, very soon? Jonathan?
Well, I mean, I think the things that are right now are the kinds of things you saw in the movie here and the kinds of things that Steve's doing with being able to augment the human's own capacity to heal in whether it's regenerating a tip of your finger or healing a certain injury better, like a diabetic ulcer or something like that. I think in the more near term, we're going to see, and there's a lot of trials going on right now in this space for tissues that perform an important role, say an occupational role, but they're not essential for life. Like, for example, a vertebral disc or a knee meniscus or something like this. This is the area that I think will be the first place where we're going to start to see some of these tissue engineering strategies, biofabrication come into play.
Dany, what about right now, what do you see using your electrodes?
Right now we see amazing possibilities. I think we're still far from it, but in some ways we may be able to skip more quickly than other types of therapies from, making a frog do it to making a human do it. And the reason for that is there are so many drugs that are already available. Some of them are over the counter, that what those drugs target are these proteins that make electrical things happen. So if you've ever taken a proton pump inhibitor for stomach acid, the proton pump is a protein, and it moves hydrogen ions, which are protons, from inside the cell to outside the cell. And that, just when I was working with tadpole tail regeneration, we found that all you had to do to get the entire tail to regenerate was give it a message, and we did that in a number of different ways, but give it the message, pump protons. And we got the entire tail. So we have drugs that you can take now and that pump protons, or they stop something. They stop your proton pumps from pumping their inhibitors. I told the story about serotonin can have this tremendous effect via its effect on the electrical status of the cells. Well, Prozac can have the same kind of effect. We have all these drugs, and their targets are these electrical signals. So in some ways, it could be very rapid because we already have the medications, but we do have a long way to go in terms of understanding for any given organ that you lose. You have heart damage. We don't know, oh, well, yeah, if you take Prilosec, your heart will grow back. We don't know that yet.
Right.
But we're getting there. It won't. So it's possible that once we really decipher that language, we can speak it, we can start going in there and fixing the grammar. And it would be potentially as easy as taking a pill.
Doris, I'll get to you last, but your day-to-day is kind of using these applications. Is that correct? Yeah.
Yeah. Yeah.
Tell us how you do that.
So we've got between 8 and 10 million patients have been treated with an extracellular matrix scaffold, but most of them are for rather ordinary, everyday things. And there's good reasons for that, like hernia repair. Instead of using a synthetic mesh made out of polypropylene, you would make it out of extracellular matrix. And these are used for that now. 20 years ago, they didn't exist. Those sorts of scaffold materials didn't exist. Same thing, it's used for the lining of the brain to replace that. So there's lots of those everyday things. And the reason those things got to the clinic so quickly was because they're regulated as a device by the FDA. They were promoted in a non-regenerative medicine approach rather than simply say, hey, look, we just got a different type of mesh material for you. So it gets out there and now got all these patients treated. One more thing, because it's an aspect of it that isn't scientific, but you might not think of in terms of why you don't see more clinical translation. When you have a regenerative medicine approach that is significantly different than the standard of care, and even though you're absolutely convinced it works, you can point to all of these studies. You go into, and you have a patient that walks through the door, and you want to use that, and you go ahead and use it. Your colleagues are looking at you saying, "What are you doing? You can't. Don't you know we've been taught how to treat these patients? Right. Aren't you? You know, this is so you've got peer pressure to say don't do it, and then you've got how are you going to get it reimbursed? Because you got a solution here, but there's no way to pay for that right now. And until you prove it, 10 years later after this money, and then on top of that, you've got lawyers standing out there waiting to sue you the first time anything goes wrong. I mean, so what are the incentives to do these? You've got your colleagues saying, no, you're not going to get paid for it, you're at risk of getting sued, and yet your patient said, please, So that's as big a barrier as a scientific barrier is for translating regenerative medicine.
And that's the heartache of being an innovator, right? Being someone who is outside the box and not in the mainstream, which probably I think all of you, maybe Jonathan a little less than others, have faced a little ostracism. Is that fair to say?
Oh, yeah.
Yeah, sure.
Yeah.
A little.
So,
Frankenstein.
Yes, of course. Right. What about you, Doris?
Well,
and then we'll talk about money, because we promised we were going to discuss that.
Yeah, the thing about regenerative medicine is it's unlike any other treatment in that when you give it, you give a drug, it's going to wear off. You give a drug, it may have an effect and one you don't. Apparently, proton pump inhibitors grow salamander tails inside you. I don't want that. But the point is, with regenerative medicine strategies, they're forever. So when you give them, you have to know they work. You have to know they're going to hang around for the long term and be safe. And you have to know that they're going to actually be flexible and change as the individual changes or faces other issues. So the burden is higher for these than any other therapy that exists. At the same time, the potential is huge. Right now, we may not be able to build a heart in a lab, but I predict within the next 2 to 3 years, we'll see a liver be built and transplanted into a patient. We will see, I guarantee you, we'll see a liver. Beyond that, pancreas is being built.
Lung lobes will get there too.
I was going to say, lung is being built. We're going to see these happen. But even today, we can build a cardiac patch. We can build new blood vessels. We can build a lot of tissues that are simple tubes. Those tubes we were talking about earlier. Bladder. There are new cosmetic ears. There are a number of strategies that already exist, and and I would go so far as to say we're not building organs and tissues, we're building hope. We're building the future and the potential for us to actually do what we're talking about here, forever young. We're building the potential to keep people healthy longer and able to function at a level that they haven't been able to before.
I think something to keep in mind too is people say, well, what are we going to do if we can solve all these problems and live to be 120 or 100? I don't want to live to be 120.
Right.
I mean, and you don't want to hear me talking about the same thing for another, X number of years. We frequently say, what?
But you want to be healthy.
Healthy is quality, not quantity.
That's right.
And I think we'd all like to live to be 80, 85, and functional, have all our faculties up until a couple days before we go, right? And then you're gone. Then that's fine. They can all go at once. Everything happens when not a single usable part left, right? But you're healthy until a week ahead of time. So it's quality, not quantity. I don't think we want to live forever.
So let's just make a couple predictions here. It's 2025. What's out there? Bob's, body parts, off the shelf, heart valves. You know, tell me what the future is, Doris. What are we going to see?
I think by 2025, if man is still alive, wasn't that a song?
If we're not, you know, trying to make it on Mars.
I really think that we're going to begin to see off-the-shelf organs and tissues we're going to solve the organ transplant problem, at least the liver and kidney and lung transplant problem. I also believe that we're going to change our endogenous repair processes so that the future is not ever getting to the point that you need that organ. We're going to learn how to use these cells earlier, use stem cells that we all have in every organ and tissue, and really maximize our endogenous capacity for repair to keep us from having to have long-term chronic disease. And then I think we will be printing. I think we will be, you know, put the four of us in a room and don't let us out for 6 months, and Steve and Dany will get those limbs built.
I was going to say, that I think that as far as the bioelectrical control, 8 years is not a long time to figure it out, but I think that the combination, once the electrical signals, we have a better sense of how they work, they will become a very important part of the bioreactor into which the personalized printed organs are going to go, or the hearts. That's going to be the first step of bringing that into it, is going to be to bring these things together.
I mean, thinking about this culture that we're talking about in 8 years, I mean, I would love it if we could see engineers becoming an integral part of the healthcare engine. So the way I see this, though, is sort of like what you were saying. If we have the printer in the OR, right? Or you're thinking about using clinical imaging to create data to inform surgical approach. Should I go this way or this way? Instead of opening up the patient and saying, well, gosh, what should I do now? You actually have the information ahead of time, and you may actually have a model of it or a hologram you can stare at. You know, I mean, we know about robotic surgery, but like, that's a big device. There's a whole lot of ways to sort of democratize this approach where every hospital has the capacity to integrate with, you know, well-trained people that can help the doctor in their approach, perhaps help them provide a better graft. I mean, so many of the medical revolutions that have happened that we know about in prosthetic industries, whatever, was doctors reaching for something around the OR because they got this thing going on with this patient, and that turned into a medical device. Well, if you have an engineer that's actually involved in that process, you could maybe have a lot more transformative components. But you know, that also comes down to, in that culture, thinking about like what Steve said in terms of reimbursement. How does that piece get paid for? I mean, I think this is a big component to thinking about the overall federal strategy of supporting research. When you try to translate this research, maybe there's a way of thinking about how citizens can get in on this new technology, but how do we share this risk? How do we share this knowledge space in a way that isn't, you know, sort of a pendulum swinging of everybody getting the latest thing and then everybody suing, and, you know, there's a potential here to put more pieces on the table.
What we don't talk about, though, and you know, is a dirty little secret in science, is we're all taught that if we don't bring in our own grants and our own money, then we're not successful. That if I work with you and I help you bring in grants, and it doesn't count for me. Right. So part of what has to change is the recognition that science is a team sport. Yeah. And institutions have to value that team mentality, or it's never going to happen.
One of the things we talked about offline before we began this evening was funding. And this, for scientists and biomedical scientists, bioengineers like yourselves, this is a very big issue. Tell me what some of your concerns are. Doris, do you want to?
Well, I mean, science funding has gone down over the last 2 decades, and about 5 years ago it was at the lowest it had ever been in my lifetime. Building one heart, one human-sized heart costs me about $200,000. And until we get it reproducible and simple, it's going to keep costing $200,000. Well, an average NIH grant is $250,000 a year. That's one heart a year. It's not going to get us there very fast.
If there's one thing you could say or do, what would it be to ensure that funding continues?
Science isn't technology. We don't say, I'm going to go in and discover that electrical signals can trigger regeneration. You go in and you spend, so that movie, the electric face movie, it took me 4 years. It was an accident. The microscope where I made the movie is a $250,000 microscope. I didn't go in one day and say, I'm going to make this movie. It's not engineering. It's not problem solving. And you can't put in a grant on this date, I will have the crucial insight. But there are some private foundations now that are doing things in a different way, and I want to shout out actually for Templeton, which has just awarded a fellowship to one of Mike Levin's postdocs to continue this frog leg regeneration study, and to the Paul G. Allen Family Foundation, which is trying a different approach, which is to pick out a person, and in this case, Mike Levin was one of those people, and bet on their creativity and their productivity, and basically say, here is all the money you need so you don't have to spend 60 to 80% of your time writing grants instead of doing experiments. Take this and see what you can come up with. And I think that's a very important and useful approach.
Okay, we're going to take a few questions from the audience. Charlotte, age 12, has, I think, a really great question. Could we improve upon nature, making larger muscles or a stronger ACL? And could you do this for an extinct species?
Oh, man.
Good question, Charlotte.
Yes, it was a good question. And we get asked that a lot because of the work we're doing with the muscle stuff. But I don't think we can ever improve upon Mother Nature. That's it. We don't even understand Mother Nature. You know, but what we can do is hopefully harness all of some of the signals that she's got to take advantage of what Mother Nature does so well. And so to make a superhuman, no, I don't think we want to do that, first of all, but I don't think we can do it. Like in the example with the salamander, why does it stop growing when it gets to here, you know, every time? Yeah. You know, and when we would actually tried some of these experiments where we knew we can grow new muscle, let's do it again, do more. And you can then temporarily, it will grow more, but then when the body doesn't need it, it regresses back to normal. It's just so smart. I mean, it just makes me realize how little we understand about biology. I wrote a perspective piece a couple of months ago called "Too Much Technology, Not Enough Biology." So the technology and the printing and such is fantastic, but if we don't know what we want to print, we can't do it. That was why my no came out so quickly. But that was a great question.
The extinct species piece. We can actually take DNA and we can actually begin to create tissues from extinct species. I'm not sure, you know, it's like we said earlier, everything has an effect you know and one you don't. I'm not sure we want to yet, but we could.
If I can just build on that, I mean, one of the idea that there is an optimal muscle is I think oversimplifying. So one of the things that evolution does, evolution has to balance competing needs. So, and for reasons we don't know yet, it may be that if a muscle gets too big, you know, your toes fall off. I mean, but there's a trade-off that we don't know about. And that if we pick out something that we happen to like and say, oh, I'm going to optimize that, you simply don't know what you're hurting at the same time.
The same muscle you use to build a fly wing that can do something phenomenal would not work in a human. I mean,
well, that leads great into the next question from Erica the Redhead. She writes, Do you think regeneration can be used to replace or fix damaged cells in cases like cancers or in autoimmune disorders?
Oh, absolutely.
Yeah. Yes.
Yeah.
Absolutely.
Yes.
The one thing stem cells do better than anything is reduce inflammation. Absolutely, no question.
Well, there's something uncomfortably close about what you guys are doing and cancer. The concept is unchecked
So what's a cancer
growth.
cell? What's a cancer cell? A cancer cell is a cell that can become a lot of different things and also that reproduces.
Right.
What's a stem cell? It's a cell that reproduces and can become a lot of different things.
Right.
The difference is a cancer cell doesn't know when to stop. And some of us believe that some tumors are actually stem cells gone bad. So I think really understanding that interaction is critical. None of us want to build that muscle that is now a tumor. Right?
Has anyone seen tumorous or cancerous unchecked growth in the labs?
Oh, sure.
Yeah.
Oh, sure.
Again,
how do you tell a cell is a true stem cell? True pluripotent can become anything stem cell. You transplant it and you watch it make a tumor. It's not a cancerous tumor, but it's a tumor. It's a teratoma.
unchecked growth.
Unchecked growth.
Teratomas are actually, they're a perfect illustration of this issue of shape being so important. So teratomas, for those of you who don't know, they are tumors that form often from the cells that make eggs, for example, the cells start to become different tissues. So, but they don't know how to get into the right shape. So what you end up with is like a blob of tissue that's growing hair. And some of its cells are producing stomach acid, and they're truly horrifying. They have teeth sometimes. The cells are following the right path to become a tissue, and it's called differentiation is the big word for it, but they are differentiating into different cell types just as their genes are telling them to do. But they have completely lost any information about what shape they're supposed to be. And so it's a blob of tissue cell types. So, but I want to go back for a second because cancer, regeneration, development, these are all, we look at all of these things from that perspective of can looking at the electrical signals teach us something that we haven't learned by looking at the chemical signals that are controlled by the genes. And another amazing thing about electricity is that we can generate tumors genetically. So we can inject a cancer-causing mutant gene, and at the same time inject the gene for a protein that will allow potassium ions in. Let potassium ions out. And what happens is the tumor doesn't form. So that potassium current changes the electrical status of the cell, and that tumor-causing gene can't form a tumor. The other thing that you can do is you can put in a gene for, there's this amazing thing called optogenetics. I'm not going to go into it right now, but into detail, but these are proteins that you turn them on and off by shining light on them. And if you make one of these tumors by injecting the mutant gene, and you also inject the gene for one of these light-activated channels, you can wait till the tumor forms, then you turn on the light, and the tumor goes away. And in frogs, this works better than most of our cancer drugs. So again, this connection between the electrical signals, the way the cells are talking to each other, in this context of generating a shape, controlling a shape, making a structure. Again, I think it's just really powerful. Go home and thank your electrical signals.
I think she brings up a good point, though, also with another opportunity for engineering, and that is to create models of patients' actual tumors, for example.
Yeah.
And instead of testing the drug's effectiveness on the patient, why don't we do it on these tumors that are the patient's tumor?
That's already happening.
Yeah, no, I agree. But like one of the new strategies that is gaining traction in a lot of different tumors is to actually differentiate it. So instead of having this cancerous tumor, turn it into a pile of bone. Well, if I have a pile of bone sitting somewhere, that's a lot better than a tumor, because now it's just sitting there as a chunk of bone. But you could test that out in a set of engineered tumors so that you wouldn't have to worry about trying it out on the patient.
All right, I got 2 more questions. Everybody wants to know, when is this heart going to happen?
Give me 5 years.
Okay, you got 5 years. And finally, and this is also, you get to pick one, which Marvel hero is more likely to be realized by research? Wolverine, who's healing, or Deadpool, regeneration? What do you pick, Jonathan?
Oh, I'm going to go Wolverine.
Okay.
Oh, I'm not that kind of nerd. Wolverine.
Okay, fine.
Wolverine.
Okay.
Deadpool.
All right. Thank you all for coming. Thank you to our amazing scientists. It was an incredible conversation.